from the final last semester and test 1, 2, 3 this semester. Okay? So there's still a fair amount on there. I think it's worthwhile to make that final exam 20% of your grade. Okay? So just make sure, start preparing for it. You know it's going to be in 2 Tuesdays. Okay? It is coming up. The next couple of chapters, after 22 and 23, 24, 25, 26, I don't bother covering those. Okay, I really don't. If you need to have a lot of information on electrolytes and digestion and nutrition, you'll have that in your nutrition course. If you don't need that, then they must not have told you to take a nutrition course. So I don't worry about those chapters at all in this class. Okay? It's not because I'm in a rush. Actually, I'm way ahead. All right, so it's not that I don't want to cover that stuff. It's just I don't think I need to cover it. We're going to jump right up to reproduction. That's how we're going to finish the semester out. After today, we have 4 lectures left. So we've got plenty of time to cover these. It looks like a lot, 3 chapters there, but the development one, I point to like 2 pictures, that's it. I spend most of the time on male and female reproduction. And you'll see they're very, very similar. So it goes pretty quickly here, and it's usually pretty interesting. So that's where we'll start in just a second. I also want to remind you our 3rd practical is this Monday. It's our last Monday. We don't have another Monday lab period because of the Easter break. So this is the end of the semester all of a sudden. And remember, it will cover the digestion lab, the urogenital lab, and the frog lab. Okay? Hey, what did you think of the frog lab? Was it the coolest lab you've ever had?
Yeah.
It's the coolest lab I've ever had. That's so cool. Even though I love dissecting bodies in gross anatomy class, I think this is cooler because it's live for a little bit. So does anybody have any questions on that lab? I've already heard some people were telling others, and they sat down with them and told them everything about it. And so that was good. I got a little report back from a former student. But any questions at all? Does everybody feel like they understood what they saw? Because hopefully it'll give you a little more understanding when you deal with those patients who are brain dead. You've got some idea. Of course, you have to be sure they're brain dead. I was sure, right? Because we cut that head completely off. You're not going to see too many like that, where their head's completely cut off. You're going to see people with mashed skulls. And then you have to wonder how much of their brain is still working. So you still have to rely on that neurologist at some point to look at those EEGs, those electroencephalograms, and tell us, is there any brain activity left in there? Okay, it's not quite as easy as my frog lab, but it's a starting point. And it's certainly a starting point most people don't have. Really, most people don't. So if you have any questions about it, please ask. I do want you to understand it and not misrepresent it to anybody. All right, great. Our practicals will be, as usual, 2 o'clock and 3 o'clock on Monday. I wanted to put a little word in here about evaluations. Because we're doing the online evaluation things now, have you all gotten emails about it? Because they don't send anything to me, which is kind of screwy. If it was up to me, I would not let you do evaluations yet. I think they opened April 3rd or something. And you know, you hadn't even had the frog lab. Most important lab we have the whole semester. So if it was up to me, I would say do evaluations during the very last week of school. So I'm just asking you, judge the whole course. Okay? Judge the whole course as usual. So I'd wait a little bit. You don't have to be in a hurry to do those. I don't know if people have started doing them or not, but that's it. Judge the whole course. Our final exam is scheduled for Wednesday, and we already decided last week that we would move it from 8 a.m. to 9 a.m. Okay? It's not going to be that much longer than a regular test, so you have plenty of time. If anybody's worried about it and they need to stay longer, it's no problem. You can stay into the lunch break. I'll be here all day. It's no problem at all. So if anybody's uncomfortable with it, just talk with me. But otherwise, we'll sleep in another hour. You can study another hour. We'll start at 9. So any questions about where we're going the rest of the semester? Because I just want to spend the last 4 lecture days just lecturing. Not worrying about anything. Just go through the reproductive system. Everybody good? Questions, comments, suggestions, complaints? None of that. All right. Wonderful. Then let's jump way up to chapter 27. The authors of this textbook decided to start with the male. I don't know if they're sexist or anything like that. I think I would probably start with the male too because it's an easier system. So it's a nice way to ease into reproduction. The female reproductive system's a little more complex. Okay? We're going to start with a general overview of development in the initial pages of this chapter. Other textbooks will have a regular development chapter and then do male versus female. They just combined it into the male here. So we're definitely going to start with this one. So we're going to go backwards a bit. This was mentioned briefly in the video that you watched, the NOVA video. But I want to go back a little bit and just hit some highlights here. So asexual versus sexual reproduction. Going way back. When we think of reproductive system in humans, we think sex. But we got to get down to that cellular level to see what's happening. And we still use that term sex. So we need to understand that. So asexual is without sex. Sexual is with sex. But at the cellular level, they really don't mean that. We don't start thinking of copulation here. We start thinking of how these cells divide. So for asexual reproduction, it's mitosis. It's regular old mitosis. What were the 4 phases of mitosis? What were the letters for them? P-mat, remember? Prophase, metaphase, anaphase, telophase. Just simple P-mat. So we went through that in pretty good detail last semester. Just want to kind of bring that back to the top of your brain for a couple of minutes here. So we talked about things that were happening in the cell. We saw how the nuclear membrane dissolved. We saw how the chromosomes condensed. They formed into those little X's for us. So that was happening during the prophase. At metaphase, those little chromosomes climbed onto the spindle in the center of the cell. So at metaphase, you had those chromosomes lined up at midline. At anaphase, those chromosomes, which were actually duplicated chromosomes, split. So we had 2 sets of chromosomes going to 2 different ends of the cell. They went all the way to the ends, and then at telophase, we just said, okay, now those chromosomes will decondense, and the nuclear membranes will reform twice. And then the cell membrane will pinch off. We included all of that in telophase, just to make it easy. So the idea there was we should start with 1 cell that had how many chromosomes in a human being? 46. And we should end with 2 cells that have how many chromosomes? 46. And they should be copies. They should be clones, okay, if everything worked right. So you see some other terms I put up here. Fission and cloning. Those are really kind of alternate terms for mitosis. Because you really are cloning. You want 1 skin cell to turn into 2 skin cells. You don't want it to differ. We don't want to all of a sudden grow a liver cell on our skin. That's not going to help us. So we want cloning. Cloning is a wonderful, great thing. You're doing it right now. We're doing it for our entire lives with most of our cells. 1 set of cells that does not continue to clone, it stops around age 6, 7, or 8, would be what type of tissue?
Nerve.
Nerve tissue. Nerve tissue seems to stop when we're fairly young. So we said, well, okay, that helps us explain why the spinal cord doesn't go all the way down our back. The spinal cord stops around that L1 region because it stopped growing then. The rest of the vertebral column, the bones kept on growing. Okay? So just reviewing everything today, going way back. So asexual reproduction simply means there's no differentiation. We're not trying to make different cells. We do not want variety. We want cloning. So no variation, and all the cells are diploid. They are the full complement. This happens in other animals and plants as well. Everybody's doing mitosis, lots and lots of cloning of cells. Alright? So, sexual reproduction. This is where it gets complicated again. When you look at it grossly versus cellularly, things get a little bit confusing. But in the end, what we want is variety and haploid cells. That's the biggest take-home message for sexual reproduction of cells. For sexual reproduction of cells. We want variety, and we want haploid cells. So if the 2 sexes, the 2 different genders, produce haploid cells, well, when they get together and have sex, not sexual reproduction, but when they have sex the way we mean it colloquially, well, then the 2 haploid cells can join and form a single diploid cell. So then you're done. There's no more sexual reproduction for a while because that diploid cell now undergoes mitosis like crazy. And it will have some variety. For that, you've got to take an embryology class to get into that. But we start with 1 fertilized egg that becomes 2, 4, 8, and so forth. Okay? So try not to confuse the act of sex that we think of with sexual reproduction. When we talk about sexual reproduction, that's what's happening in your ovaries or your testes. And that's it. That's it. And we're trying to make cells that vary, and they're only haploid. So in the guys, we're trying to make sperm, and the ladies, we're trying to make ovum. And if they've been made properly, all the different ova and all the different sperm should be different from each other. They are not clones. And they should have half the normal number of chromosomes. Have I beat that to death? Everybody with me there? All right. We said the name for that type of division is meiosis. Meiosis. M-E-I-O-S-I-S. Meiosis. We'll get into that in the next couple of lectures. We'll actually look at meiosis in a little more detail. We'll compare it to that mitosis. It's very similar. We're going to look for 2 little differences in the guys, 3 little differences in girls. But otherwise, we're still going to use prophase, metaphase, anaphase, telophase. Okay, we'll still use those same things. Right? Okay, so we know what's going to happen at the cellular level. It's a little preview. We will come back to that. In our broad overview here, the textbook on pages 1035, 6, and 7, it discusses primary versus secondary sexual characteristics. Well, primary characteristics is very simple. The primary thing that makes men different from women is their gonads. That's the primary sexual characteristic. Do you have testes or do you have ovaries? That is the primary difference. The secondary differences are generally all those things that develop at puberty. That's the secondary sex characteristics. So you know this. You go to a school, you go to a daycare or something, and you look at little 2-year-old, 3-year-old, 5-year-old kids. The boys and girls aren't very different. They may act different, because we hate boys. But as far as their physical appearance, they're really not all that different. But then in puberty, now the secondary sex characteristics develop. So we know that the women are going to develop breasts. They develop usually a widening of the hips. The boys are going to develop facial hair. They're going to have a larger growth spurt than the ladies even. Both will have growth spurts. Hair on the face. Hair in the armpits. All of those things are secondary sex characteristics. So anything that you think of that's male or female that's not the gonads is a secondary sex characteristic. We know at the cellular level, we can be even more specific. Because we know of the 46 chromosomes that you have in all of your cells, almost all of your cells, that the last 2 pairs are what we refer to as the sex chromosomes. So if we inherit one X from dad and one X from mom, we produce a little baby girl. 2X. So we went over this briefly when we were doing the Punnett squares. And we did sex-linked characteristics. Okay? Well, we were linking them to these particular genes. To the genes on these chromosomes, the 23rd pair of chromosomes. We know if dad has a sperm that has a Y chromosome, and the egg is an X, now we're going to produce a little boy. Simple as that. So really, when you talk about primary versus secondary sex characteristics, that's gross. That's stuff you can see with the naked eye. You can't always see the gonads. You might have to cut somebody open, but you can still see with the naked eye. But when we get down to the cellular level, it's at the chromosomes. Right, that's where we determine sex. And if we've got a pair of X's, it's female. A pair of Y's, it's male. Okay? So 2 different ways of looking at it there. When we look at what's going on with the guys in particular, that Y chromosome is the difference, right? Because both sexes have an X. But when we have 2 X's, it's female. When we have a Y and an X, it's going to be a male. That's because there's a gene on that chromosome. It's called the SRY gene. I don't really care that you know that. But that gene helps produce a protein called TDF, testis determining factor. And so during development, in the very, very early stages, we take this very primitive gonad and we start to change it just a little bit into a testes instead of an ovary. So some people look at it this way, is they think that female is sort of the default. I've heard some people, especially after they take a psychology class, they'll say, we all start female. Okay, well, that's 100% wrong. Okay? We all start right here when a sperm meets an egg. And if the sperm is an X, then we start female. And if the sperm is a Y, we start male, period. But I think as somebody who hasn't looked very much at the cellular goings-on or the anatomy really, is we start with this.